Welcome back to Chapter 3 of Concepts of Biology, Cell Structure and Function. Section 1, How Cells Are Studied. By the end of this section, you will be able to describe the roles of cells in organisms, compare and contrast light microscopy and electron microscopy, summarize the cell theory. Introduction. Close your eyes and picture a brick wall. What is the basic building block of that wall? It is a single brick, of course. Like a brick wall, your body is composed of basic building blocks, and the building blocks of your body are cells. Your body has many kinds of cells, each specialized for a specific purpose. Just as a home is made from a variety of building materials, the human body is constructed from many cell types. For example, epithelial cells protect the surface of the body and cover the organs and body cavities within. Bone cells help to support and protect the body. Cells of the immune system fight invading bacteria. Additionally, red blood cells carry oxygen throughout the body. Each of these cell types plays a vital role during the growth, development, and day-to-day maintenance of the body. In spite of their enormous variety, however, all cells share certain fundamental characteristics. A cell is the smallest unit of a living thing. A living thing, like you, is called an organism. Thus, cells are the basic building blocks of all organisms. In multicellular organisms, several cells of one particular kind interconnect with each other and perform shared functions to form tissues. For example, muscle tissue, connective tissue, and nervous tissue. Several tissues combine to form an organ, for example, a stomach, heart, or brain, and several organs make up an organ system, such as the digestive system, circulatory system, or nervous system. Several systems functioning together form an organism, such as an elephant, for example. There are many types of cells, and all are grouped into one of two broad categories, prokaryotic and eukaryotic. Animal cells, plant cells, fungal cells, and protist cells are classified as eukaryotic, whereas bacteria and archaea cells are classified as prokaryotic. Before discussing the criteria for determining whether a cell is prokaryotic or eukaryotic, let us first examine how biologists study cells. Microscopy. Cells vary in size. With few exceptions, individual cells are too small to be seen with the naked eye, so scientists use microscopes to study them. Aside, one cell that is potentially viewable with the naked eye, one human cell, is the human egg. A microscope is an instrument that magnifies an object. Most images of cells are taken with a microscope and are called micrographs. Light microscopes. To give you a sense of the size of a cell, a typical human red blood cell is about 8 millionths of a meter, or 8 micrometers, in diameter. The head of a pen is about 2 thousandths of a meter, millimeters, in diameter. That means that approximately 250 red blood cells could fit on the head of a pen. The optics of the lenses of a light microscope changes the orientation of the image. A specimen that is right side up and facing right on the microscope slide will appear upside down and facing left when viewed through a microscope, and vice versa. Similarly, if the slide is moved left while looking through the microscope, it will appear to move right, and if moved down, it will seem to move up. This occurs because microscopes use two sets of lenses to magnify the image. Due to the manner in which light travels through the lenses, this system of lenses produces an inverted image. Binoculars and a dissecting microscope work in a similar manner, but include an additional magnification system that makes the final image appear to be upright. Most student microscopes are classified as light microscopes. Visible light both passes through and is bent by the lens system to enable the user to see the specimen. Light microscopes are advantageous for viewing living organisms, but since individual cells are generally transparent, their components are not distinguishable unless they are colored with special stains. Staining, however, usually kills the cells. Light microscopes commonly used in the undergraduate college laboratory magnify up to approximately 400 times. If you're in a microbiology lab, that will probably be up to approximately 1,000 times. Two parameters that are important in microscopy are magnification and resolving power. Magnification is the degree of enlargement of an object. Resolving power is the ability of a microscope to allow the eye to distinguish two adjacent structures as separate. The higher the resolution, the closer the two objects can be, and the better the clarity and detail of the image. When oil immersion lenses are used, magnification is usually increased to 1,000 times for the study of smaller cells like most prokaryotic cells. Because light entering a specimen from below is focused onto the eye of an observer, the specimen can be viewed using light microscopy. For this reason, for light to pass through a specimen, the sample must be thin or translucent. A second type of microscope used in laboratories is the dissecting microscope, which is on the right. These microscopes have a lower magnification, 20 to 80 times the object size, than light microscopes and can provide a three-dimensional view of the specimen. Thick objects can be examined with many components in focus at the same time. These microscopes are designed to give a magnified and clear view of tissue structure as well as the anatomy of the whole organism. Like light microscopes, most modern dissecting microscopes are also binocular, meaning that they have two separate lens systems, one for each eye. The lens systems are separated by a certain distance and therefore provide a sense of depth in the view of their subject to make manipulations by hand easier. Dissecting microscopes also have optics that correct the image so that it appears as if being seen by the naked eye and not as an inverted image. The light illuminating a sample under a dissecting microscope typically comes from above the sample, but may also be directed from below. As you can see in this image, this pictured dissecting scope can do both. You can have your bottom light and your top light. Each intensity should be separately controllable so that you can alter your view of your specimen. Electron microscopes. In contrast to light microscopes, electron microscopes use a beam of electrons instead of a beam of light. Not only does this allow for higher magnification and thus more detail, it also provides higher resolving power. Preparation of a specimen for viewing under an electron microscope will kill it. Therefore, live cells cannot be viewed using this type of microscopy. In addition, the electron beam moves best in a vacuum, making it impossible to view living materials. In a scanning electron microscope, a beam of electrons moves back and forth across a cell's surface, rendering the details of cell surface characteristics by reflection. Cells and other structures are usually coated with a metal like gold. In a transmission electron microscope, the electron beam is transmitted through the cell and provides details of a cell's internal structures. As you might imagine, electron microscopes are significantly more bulky and expensive than are light microscopes. And in this image here, you have a comparison of a light microscope of a stain of some kind of a tissue sample, apparently. The tiny dots there in the left panel are the salmonella cells, while on the right, that's salmonella close-up with a scanning electron micrograph, which has been colorized. Those aren't the colors that you would see that structure in nature. Careers in action. Saito Technologist. Have you ever heard of a medical test called a Pap Smear? In this test, a doctor takes a small sample of cells from the uterine cervix of a patient and sends it to a medical lab where a Saito technologist stains the cells and examines them for any changes that could indicate cervical cancer or a microbial infection. Saito technologists are professionals who study cells through microscopic examinations and other laboratory tests. They are trained to determine which cellular changes are within normal limits or are abnormal. Their focus is not limited to cervical cells. They study cellular specimens that come from all organs. When they notice abnormalities, they consult a pathologist who is a medical doctor who can make a clinical diagnosis. Saito technologists play a vital role in saving people's lives. When abnormalities are discovered early, a patient's treatment can begin sooner, which usually increases the chances of successful treatment. Cell theory. The microscopes we use today are far more complex than those used in the 1600s by Antony van Leeuwenhoek, a Dutch shopkeeper who had great skill in crafting lenses. Despite the limitations of his now ancient lenses, van Leeuwenhoek observed the movement of protists, a type of single-celled organism, and as mentioned before, it would be a eukaryote, and sperm, which he collectively termed animalcules. In a 1665 publication called Micrographia, experimental scientist Robert Hooke coined the term cell, from the Latin cella, meaning small room, for the box-like structures he observed when viewing cork tissue through a lens. In the 1670s, van Leeuwenhoek discovered bacteria and protozoa. Later advances in lenses and microscope construction enabled other scientists to see different components inside cells. By the late 1830s, botanist Matthias Schleiden and zoologist Theodor Schwann were studying tissues and proposed the unified cell theory, which states that all living things are composed of one or more cells, that the cell is the basic unit of life, and that all new cells arise from existing cells. These principles still stand today. That's the end of Section 1 of Chapter 3. Next time we'll come in with Section 2, comparing prokaryotic and eukaryotic cells. Until then.